Hola, están my compañeros. I would hope that you can notice a couple things right in front of you real quick. One is the NIH logo. Two is the title, a SARS-like cluster of circulating bat coronaviruses shows a potential for human emergence. When you read that, you think of a naturally occurring process. And then three, I would like for you to notice the date, 2015, December. It's about 1 year after the moratorium on gain-of-function research. This article has to do with them creating a chimeric virus. They took that circulating bat virus and they made it into something even more virulent. And that's what the article is about. And I can prove that they knew that it was GOF research after the moratorium, and they say they did it because they didn't expect for it to be virulent. But here, I'll read what it says in the abstract. It says, here we examine the disease potential of a SARS-like virus, SHC-014 coronavirus, which is currently circulating in Chinese horseshoe bat populations. So that's the virus that the title mentions. But then using the SARS coronavirus reverse genetic system, we generated and characterized a chimeric virus. So this is an unnatural process. It's a chimeric virus expressing the spike of the bat coronavirus, so it's half bat coronavirus, in a mouse-adapted SARS coronavirus backbone. So they injected it into a mouse, which was genetically altered in order to be similar to our own system, to see if it would infect our system. More specifically, the angiotensin converting enzyme ACE2 receptors, which are located on your little cilia, which are the hairs of your throat. They're the hairs of your throat. They work all your spit up. They're like little arms that keep moving. But back to what I was saying. So using the SARS coronavirus reverse genetic system, we generated and characterized a chimeric virus. It expresses the bat coronavirus spike, SHC-014, and it expresses it in a mouse-adapted SARS coronavirus backbone. The results indicated that the group 2B viruses encoding the SHC-014 spike in a wild-type backbone can efficiently use multiple orthologs of the SARS receptor human angiotensin converting enzyme 2. That's saying that they injected it and it was effective. It can replicate efficiently in primary human airway cells and achieve the in vitro titers, which it means that it grows inside of them. Vitro titers equivalent to epidemic strains of SARS coronavirus. So I will leave all the links. I'm going to be searching around a bit, but go ahead and listen to Dr. Fauci's refusals. This is the National Institute of Health. Baric, this is an article written by Baric and Shi Zhang-li of the Wuhan.
We don't know whether the pandemic started in a lab in Wuhan or evolved naturally, but we should want to know. 3 million people have died from this pandemic, and that should cause us to explore all possibilities. Instead, government authorities, self-interested in continuing gain-of-function research, say there's nothing to see here. Gain-of-function research, as you know, is juicing up naturally occurring animal viruses to infect humans. To arrive at the truth, the U.S. government should admit that the Wuhan Virology Institute was experimenting to enhance the coronavirus's ability to infect humans. Juicing up superviruses is not new. Scientists in the U.S. have long known how to mutate animal viruses to infect humans. For years, Dr. Ralph Baric, a virologist in the U.S., has been collaborating with Dr. Shi Zhang-li of the Wuhan Virology Institute, sharing his discoveries about how to create superviruses. This gain-of-function research has been funded by the NIH. The collaboration between the U.S. and the Wuhan Virology Institute continues.
The article that I'm going through in the bottom half of the screen is the exact same article that it was just titled, that says SARS coronavirus might move out of bats or something along those lines. And you can see that that article is directly drawn upon information, data, resources of the Wuhan Institute of Virology. Chapel Hill and Wuhan Institute of Virology were making chimeric viruses, and the National Institute of Health knew all about it. Let's see, gain of function.
Insert bat virus spike protein into the backbone of the deadly SARS virus, and then use this man-made supervirus to infect human airway cells. Think about that.
This is exactly what it says in the bottom half of the screen. It says, suggesting additional functional testing was required for verification. Okay, first it's going to say, first they talk about okay, here, first they talk about a failure. It says, in the middle of the screen, suggested that the SHC04 spike is unable to bind to the ACE2. So they say, oh, it won't bind to the ACE2. And then they say, however, similar changes in related SARS coronavirus strains has been reported to allow ACE2 binding, suggesting that additional functional testing was required for verification. Therefore, we synthesized some more of the bat spike in the context of the replication component, mouse-adapted SARS coronavirus backbone. We hereafter refer to this chimeric virus that we just created as SHC014MA15. And to maximize the opportunity for pathogenesis in our new chimeric viruses that we just created in 2015 after the moratorium on gain-of-function research, in order to maximize the opportunity for pathogenesis and vaccine studies in mice. Oh, that's why they say that they did that. We made our chimeric virus to maximize our opportunity for pathogenesis and vaccine studies in mice. Despite predictions from both structure-based modeling and pseudotyping experimental, SHC014MA15 was viable and replicated in high titers, so it lives, what we created lives. Similar to the real SARS, the chimeric virus that we just created also required a functional ACE2 cilia hair on your throat molecule for entry, and it could use the human civet and bat ACE2 orthologs, just like the original bat virus, but it's not the original bat virus because it's a chimeric virus that we had just created post-moratorium to test the ability of the new virus spike to mediate or to test the ability of the bat spike to mediate infection of the human airway. In order for it to see if it would instigate, we examined the sensitivity of the human epithelial airway cells in line Calu to. That means that they took a line of airway cells, epithelial cells, and they infected it and found robust replication. They found a robust chimeric replication, robust, comparable to that of the real SARS. And to extend these findings, to extend the findings to see what more we had to find out about our new chimeric virus, we took primary airway cells, cultures, and infected and we took real human cells and showed robust replication of both the viruses. Together, the data confirms the ability of our new SHC spike to infect human airway cells and underscores the potential threat if our chimeric virus happens to get out. This is Dr. Fauci's denials.
That for a moment. The SARS virus had a 15% mortality.
And
we're fighting a pandemic that has about a 1%.
They just said that the chimeric virus that they created was as lethal as SARS urbani. And if you'll see, once again, they know that they're doing gain-of-function research, as you'll see in the article itself. In addition to offering preparation against future emerging viruses, so in addition to giving us, in addition for showing us how bad it could be, this approach must be considered in the context of the U.S. government-mandated pause on gain-of-function studies. On the basis of previous models of emergence, the creation of this chimeric virus, such as the one we just made, it was not expected to increase pathogenicity, although the virus that we just made is relative to parental mouse adapted, although it's similar to the SARS coronavirus. Similar studies examining the pathogenicity of coronaviruses, the wild type or Bonnie spike within the MA15 backbone, that showed no weight loss in mice and reduced viral replication. And so we didn't imagine that if we took the lethal SHC014 spike and we mixed it with coronavirus, we didn't know that that shows a gain in pathogenesis. On the basis, they say, and then they say, oh, since we just figured out that we created this chimeric virus, it says, on the basis of these findings, the scientific review panel may deem similar studies that we just did, like the one we just did during the moratorium, similar studies building chimeric viruses based on circulating strains, too risky to pursue. An increased pathogenicity in mammalian models cannot be excluded. So if you read exactly what that means that they're saying, oh, we did not intend to create this ultra lethal virus. We're sorry, we won't do it again.
Mortality. Can you imagine if a SARS virus that's been juiced up and had viral proteins added to it, to the spike protein, if that were released accidentally?
And what he just said is that SHC014 is the chimeric virus.
Of the lab in Wuhan.
Senator Paul, with all due respect,
listen to him.
You are entirely and completely incorrect.
This is a lie.
That the NIH has not ever and does not now fund gain-of-function research. He kind of said a double negative. In the Wuhan Institute.
Do they fund Dr. Baric?
He kind of said has not ever, almost like he was like they have not funded it. And now he's about to ask him directly about what I just have been reading to you for the last 5 minutes.
We do not fund.
Do you fund Dr. Baric's gain-of-function research?
Dr. Baric does not doing gain-of-function research, and if it is, it's according to the guidelines, and it is being conducted in North Carolina.
You don't think inserting a bat virus spike protein that he got from the Wuhan Institute into the SARS virus is gain of function?
That is not.
You would be in the minority because at least 200 scientists have signed a statement from the Cambridge Working Group saying that it is gain of function.
Well, it is not. And if you look at the grant and you look at the progress reports, it is not gain of function, despite the fact that people tweet that.
So do you still support sending money to the Wuhan Virology Institute?
We do not send money now to the Wuhan Virology Institute.
Do you support sending money? We did, under your tutelage. We were sending it through EcoHealth. It was a sub-agency and a sub-grant. Do you support the money from NIH that was going to the Wuhan Institute?
Let me explain to you why that was done. The SARS-CoV-1 originated in bats in China. It would have been irresponsible of us if we did not investigate the bat viruses and the serology to see who might have been infected.
Or perhaps
he didn't. What he is saying that they did, comparable to what I just have read to you throughout the paper, it's a lie. He is lying. He is committing perjury.
In China, responsible to send it to the Chinese government that we may not be able to trust with this knowledge and with this incredibly dangerous viruses. Government scientists like yourself who favor gain-of-function research.
I don't favor gain-of-function research in China. You are saying things that are not correct.
Government defenders of gain-of-function, such as yourself, say that COVID-19 mutations were random and not designed by man. But interestingly, the technique that Dr. Baric developed forces mutations by serial passage through cell culture that the mutations appear to be natural. In fact, Dr. Baric named the technique the no-see-em technique because the mutations appear naturally. Nicholas Baker in the New York Magazine said nobody would know if the virus had been fabricated in a laboratory or grown in nature. Government authorities in the U.S., including yourself, unequivocally deny that COVID-19 could have escaped a lab. But even Dr. Shi...
What's interesting is what I just highlighted on the bottom half of the screen says this paper that just said that they made a gain-of-function chimeric virus out of the natural virus that they were speaking about. They didn't test how virulent the natural virus is. That's not what they did. They made a chimeric virus that was absolutely deadly. And I don't know what they did with it. But regardless, it says that the NIH said a continuation of these studies was requested and has been approved by the NIH. Continuation of these studies was requested and has been approved by the NIH.
In Wuhan wasn't so sure. According to Nicholas Baker, Dr. Shi wondered, could this new virus have come from her own laboratory? She checked her records frantically and found no matches. That really took a load off my mind, she said. I had not slept for days. The director of the gain-of-function research in Wuhan couldn't sleep because she was terrified that it might be in her lab. Dr. Baric, an advocate of gain-of-function research, admits the main problem that the Institute of Virology has is the outbreak occurred in close proximity. What are the odds? Baric responded, could you rule out a laboratory escape? The answer in this case is probably not. Will you, in front of this group, categorically say that the COVID-19 could not have occurred through serial passage in a laboratory?
I do not have any accounting of what the Chinese may have done, and I'm fully in favor of any further investigation of what went on in China. However, I will repeat again, the NIH and NIAID categorically has not funded gain-of-function research to be conducted in the Wuhan Institute of Virology.
You support it in the U.S. We have 11 labs doing it, and you have allowed it here. We have a committee to do it, but the committee has granted every exemption. You're fooling with Mother Nature here. You're allowing superviruses to be created with a 15% mortality. It's very dangerous. I think it was a huge mistake to share this with China, and it's a huge mistake to allow this to continue in the United States. And we should be very careful to investigate where this virus came from.
I fully agree that you should investigate where the virus came from. But again, we have not funded gain-of-function research on this virus in the Wuhan Institute of Virology, no matter how many times you say it,
there was research.
It's so interesting because the National Institute of Health is a very large proponent of Shi Zhang-li. It's obvious that they've given her grants and given her money, and she's worked out of the Wuhan Institute of Virology. So, I mean, it probably is not that difficult to make some connections that it might be a shell game, like they just move it around a bit, but regardless, so that's those are the lies of Fauci.